where we stopped. I have already discussed with you all these questions from the previous lessons, and we stopped at plasma membrane. So we'll start our revision now from plasma membrane. Okay, so let's not waste much time and let's get into our revision session. I hope you're ready for it now. Okay, so we start with the lesson plasma membrane, and the question that will come is from page number 43, information from the text. On page number 43, they are talking about the plasma membrane structure. So most of the molecules in the plasma membrane are lipids. If this is the structure of the plasma membrane, if you will have a look at the structure of plasma membrane, it is a phospholipid bilayer. You can see we'll be discussing the detailed structure now. So they are lipids. Lipid is the important. That's why you have studied in the previous lesson that lipid forms barriers because it is an important molecule which is forming the plasma membrane. What are lipids? Actually, they consist of glycerol and 3 fatty acids. Any lipid molecule has a glycerol and 3 fatty acids. Now what happens here, that one of the fatty acid is replaced by a phosphate group. So one fatty acid replaced by, so it has a glycerol, 2 fatty acids and 1 phosphate group. So we call it now a phospholipid. So the name changes, we make it we call it phospholipid because it has a phosphate group. I hope that you understand. Then a phospholipid is a molecule that has a glycerol backbone, 2 fatty acid chains, and a phosphate containing group. We call it a phospholipid. Now, this is one molecule. In plasma membrane, there are 2 phospholipid layers which are together forming the plasma membrane. So it is a bilayer. Bilayer means 2 layers. 2 layers of what? 2 layers of phospholipids. So it is composed of phospholipid bilayer. So you can see in this figure there are 2 layers. One on the top and one at the bottom. 2 layers of phospholipids. With 2 layers phospholipids, and they are arranged how? Tail to tail. So the this is a phospholipid molecule. Here you can see this is one molecule of phospholipid. It has the blue color thing, a round structure, that is the polar head, and it has 2 non-polar tails. So each phospholipid molecule has a polar head and a non-polar tail. So 2 layers, they are arranged how? The tail to tail. So the polar heads are both facing on the outside, and inside there are the non-polar tails. Tail to tail in the plasma membrane. Phospholipids arrange themselves in a way that allows the plasma membrane to exist in a watery environment. The reason is that why they arrange like that, so that plasma membrane can exist in a watery environment. It does not get spoiled in a watery environment. So that's the outside of the cell here and the inside of the cell. And this is the plasma membrane. Yeah, you can understand it. It's like the phospholipid bilayer. You can see this is one molecule of phospholipid, a polar head and non-polar tails too. They are arranged in such a line. One layer has the head here and inside they are the tails. The other layer is upside down. The head is at the bottom and the layer. So the in between the tails are there. So these are the 2 layers. That's why we call it a bilayer. So when you're seeing it is single molecule, and bilayer means 2 layers of phospholipids. Okay. Now what are the structures that are present on the plasma membrane? So here you see there is membrane protein. This is a membrane protein. You can identify it by this structure, this membrane protein. It also communicates with outside. So then here, without this membrane protein, just like that, this is the carbohydrate chain. This big structure is a transport protein. So here you have membrane protein, here you have transport protein, and these little structures in between the tails, they are the cholesterol. So So this is a phospholipid. When they are marking like this, it's a phospholipid bilayer. These are the polar heads here and here. All the blues, they are the polar heads. And the greens are the non-polar tails. Okay, so this is the polar head, non-polar tail. This is the inside of the cell and this is the outside of the cell. It looks like a sandwich with polar heads facing outside and non-polar tails facing inside. So question is about the structures, information from text. Together, the phospholipids in a bilayer create a sea in which other molecules can float, like apples floating. So it is like other molecules can float in it, just like apples floating in a barrel of water. So we call it a fluid mosaic model. So plasma membrane is a fluid mosaic model because it can float and all the structures are like together. The phospholipids can move sideways. They can move sideways within the membrane, just as apples move around the water. Supposing you have water and you put lots of apples, the apples can move the same way the phospholipids can move. At the same time, other components in the membrane, such as proteins, also move among the phospholipids. So the proteins are also moving in between because there are different substances in the plasma membrane. A pattern or mosaic is created on the surface. So there is a pattern on the surface. You can see this pattern. The components of the plasma membrane are in constant motion, sliding past it. That's why we call it a fluid mosaic model. What is the function of the plasma membrane? It maintains balance in an organism's internal environment or homeostasis. Homeostasis is the balance in internal environment, and it is very important for the survival of the cell. One of the structures that is responsible for homeostasis is plasma membrane. So one function of plasma membrane is to maintain homeostasis. The plasma membrane is thin, flexible boundary. It's a flexible boundary between cell and the environment that allows nutrients. It allows nutrients to come in and out of the cell. Waste goes out, nutrients come in, and other products to leave the cell. All prokaryotic cells and eukaryotic cells that you've just studied in the previous lesson, that all prokaryotes and eukaryotes have a plasma membrane. It's found in everything. And this plasma membrane can separate them from the watery environment in which they exist. Because they're existing in a watery environment, so this can separate them from the watery environment. They're talking about selective permeability. What is selective permeability? This is an important property of plasma membrane. Selective permeability. What does that mean? It means that the membrane is allowing some substances to pass through, while it does not allow some substances to. So it controls what is going in and it controls what is going out. So it is allowing some substances to pass through and keeping others out. Just like a fish net, as an analogy of selective permeability. What happens? The fish are inside the net, but the water can move, the other substances can move, but not the fish. So this is selective permeability. It has holes which allow water and other substances in the water to pass through, but not the fish. Selective permeability of the plasma membrane is shown there. Like the arrows here, you can see, I'll just come to it. The arrows show that substance enter and leave the cell through the plasma membrane. It controls of how, when, and how much of these substances can enter or leave the cell. So if they give you this, what feature of the plasma membrane is this showing? So this is selective permeability. Sometimes they show you this and they ask you, or they show you what is showing by this. Which characteristic of plasma membrane is seen by this? It is selective permeability. The fish net selectively captures fish while allowing water and other debris to pass through. Plasma membrane is selective for substances entering, like oxygen can come in, glucose can come in, waste go out, carbon dioxide go out, water can go in and out of the plasma membrane. Selective permeability. Demonstrate understanding. Question 1, page 46. Identify the molecules in the plasma membrane that provide basic membrane structure, cell identity, and membrane fluidity. So which one is that which is giving it a fluidity? That is cholesterol. Basic membrane structure. Who is forming the structure? Phospholipids. Cell identity, proteins and carbohydrates. Membrane fluidity, cholesterol. So these are the different molecules of the plasma membrane which are making. Structure is being given by phospholipids. Identity of the cell is by proteins and carbohydrates that are present. Membrane fluidity by cholesterol. Remember these. What are the functions of the different molecules in the plasma membrane? How the term fluid and mosaic describe the plasma membrane. Fluid, it means that proteins and cholesterol float in the membrane. Mosaic, because it has many parts and it is forming a pattern on the surface of the plasma membrane. That's why it is mosaic. Then we come to the next part, which is about next lesson, which is about diffusion and cellular transport. Diffusion and osmosis. What is diffusion? What is osmosis? And what are the roles in the cell? Firstly, we should know what is diffusion. Just like if something is being cooked in the kitchen, the smell reaches us. How does it reach us? Because the particles move in the particles, between the particles of the air, and they reach us. So the same way, this is diffusion. Diffusion is the random motion of particles from a high concentration to a low concentration. They always move from where there are too many to the place where there are less. Like if you spray perfume, then where you are spraying, the concentration is high. So they will move from there to where the concentration is less. So the particles of gases, liquid and solids, they are in random motion. So they move. And you know that they are in random motion, which is called Brownian motion. This motion causes diffusion. Diffusion is what? It is the net movement of particles from an area where there are many particles of a substance that is concentrated to an area where there are fewer particles, that is less concentration, low concentration of the substance. So they are moving from an area of high concentration to an area of low concentration. It's a net movement. The amount of substance, that means they're moving in both ways, but more are moving in one direction, from high to low. Diffuse from area from high concentration to low concentration. So where the particles are more, we call it concentration. Concentration is the amount of substance. So when they are more, high concentration, less, low concentration. Here figure 8 is showing you the process of diffusion. Additional energy input is not required. We don't need to give any extra energy. It does not need any energy. It's passive transport. You know, there are two types of transports, passive and active. For active, energy has to be supplied. For passive, no energy is needed. The particles are already in motion. If you drop blue and yellow ink into a container of water at the two ends, after some time you'll see that it gets mixed. Why? By the process of diffusion. They just mix, like here. Diffusion, we put a drop of blue here, a drop of yellow here. Diffusion begins. They move from blue has high concentration. It will start moving this side towards low concentration. Yellow has high this side. It will start moving in the other direction. That is low concentration. And slowly they will start mixing until when they will mix? Till all the concentration becomes uniform. So diffusion causes the inks to move from high ink concentration to low ink concentration until the color becomes evenly blended in water. Given more time, the ink particles continue to mix and in the case continue to form the uniform mixture. Mixing continues till when? Until the concentrations of yellow and blue ink are same. After mixing is complete, the particle they will not stop moving. They will continue movement, but the movement will be the same in both directions. How many particles are moving from one side to the other? The same amount of particles moving from the other side to the first side. So this condition where there is no net movement, no overall change. Movement is going on, but there is no change. That is known as dynamic equilibrium. So here when you see that it has mixed together, now this has reached dynamic equilibrium. One of the key characteristics of diffusion is the rate at which diffusion takes place. There are three main factors that are affecting the rate of diffusion. This they ask. What are the three factors? Concentration, if the concentration is different, it will move till the concentration becomes equal. Temperature. When temperature increases, diffusion rate increases. And also pressure. So these are the three factors. When concentration is high, diffusion occurs more quickly because there are more particles to collide and bounce off one another. When temperature or pressure increases, again the number of collision increases. So it increases the rate of diffusion. Higher temperature, particles move faster. Higher pressure, the particles are closer together. In both cases, more collisions occur and diffusion is faster. The size and charge of the substance also affects the rate of diffusion. So mainly three: concentration, temperature and pressure, but also the size and charge can affect the rate of diffusion. Now, what is facilitated diffusion across a plasma? This you understood, right? And another question about this only. In addition to water, cells need certain ions and small molecules. They don't only need water, they need ions, they need small molecules. Like they need chloride ions, sugars to perform functions. Water can diffuse. Now, this was asked once. Water can go easily through the plasma membrane, diffuse from high to low, low to high, high to low, high to low, they will go. But the other substances like molecules, sugar molecules, or ions, they cannot move through the plasma. So how do they go? They go through proteins. They cannot move through plasma membrane. Another form of transport is facilitated diffusion. It is diffusion, high to low, but it cannot go through plasma. It needs something to go through. It needs a way to go through. So they use transport proteins to move ions and small molecules. So when they are using proteins, then we call it facilitated diffusion. By this method, substances move into the cell through a water-filled transport protein called channel protein. So which protein is allowing them to go? Channel. It will open and close and the substances can go through. There is one more type of transport protein which is called carrier protein. It can also help substances diffuse across the plasma membrane. What is the difference between the two? Carrier proteins, they change shape. Channel proteins are the same. They just like open, close. They don't change shape. Carrier proteins change shape as the diffusion process continues so that the particles can move through. Diffusion of water and facilitated diffusion of other substances require no extra energy because the particles are moving from high concentration or they're moving along the concentration gradient. We use the word gradient. So this is known as passive transport. They don't need extra energy. Okay. Now here you can see this is passive transport. Water molecule, they're just moving. They're going from high to low. These yellow dots are representing water molecule. So just moving through the plasma, high to low, diffusion of water. No energy needed. This is concentration gradient, means from high it is going to low. This is the concentration gradient. So no energy is needed. Now here, the sugar or the ions want to go, but they cannot go through here because they are big enough. So they use the channel protein to go. The channel protein will open and allow them to go. But they are moving from high to low. So this is also diffusion, but it is facilitated diffusion. Now this is carrier protein. It changes shape. It opens, takes the sugar molecule or the ion, and then it opens on the other side and the sugar molecule goes in. So channel protein, carrier protein. Both are doing what? Facilitated diffusion. Although water moves freely through plasma membrane, other substances cannot pass through it. The phospholipid bilayer, channel proteins and channel proteins and carrier proteins are kinds of transport proteins that help ions and small molecules to move across the membrane in a process called facilitated diffusion. So diffusion is simple moving. Facilitated is with the help of a facility. What is that facility? The facility is proteins. They move with the help of transport proteins. Okay, now I've already told you about dynamic equilibrium. I already told you that it will continue to move till a condition comes where there is continuous movement but no change. Overall change. Movement carries on, but no change. Both sides, same movement. That is dynamic equilibrium. Then I also told you about, they always move here. Then I told you about the factors. that are affecting diffusion, concentration, temperature, and pressure. These affect diffusion. They will increase the diffusion. Temperature is more, concentration is more, pressure is more, so the rate of diffusion will increase. Then across plasma membrane, also I explained you in the previous, this is facilitated when it is using protein. Actually, this has been asked in 2 questions. In question number 8 here about diffusion, and then they have also asked in question number 29. That's why this same slide or same text has been put on both the slides. Okay. So here the same figure I have already explained you. Let's move further. Then again in question number 9, they're talking about the same diffusion, but they're talking about diffusion of water alone through a selectively permeable. So when water is diffusing through a selectively permeable membrane, we give it a special name. We call it osmosis. It is diffusion, but it is through a selectively permeable membrane. Like in this case, for example. Here, this is diffusion, no selectively permeable membrane, but this is diffusion. But when it is taking place through a selectively permeable membrane, here, this is an example of osmosis. So osmosis is a special type of diffusion. Water molecules are only passing. Water molecules pass freely into and out of the cell through aquaporins. They are channel proteins embedded in the plasma membrane composed of tiny holes and selectively allow small changes molecules such as water to pass through. How does it work? Solution, a substance called solute is dissolved in a solvent. We discussed this earlier. Water is the solvent in the cell and its environment. Concentration is the measure of the amount of solute dissolved in the solvent. What is concentration? How much solute has been dissolved in the solvent? The concentration of solution decreases when the amount of solvent increases. If supposing more water is increases, concentration will decrease. Water goes out, concentration will increase. So the solute is not moving. What is moving is the solvent. In osmosis, only the solvent. Water is moving. It shows a U-shaped tube containing solutions with different sugar concentrations separated by a selectively permeable membrane. What will happen if the solvent can pass through the membrane but the solute cannot? Water molecules diffuse towards the side with the greater sugar concentration. The right side, the water moves to the right side as the concentration of sugar solution decreases. The water continues to diffuse until dynamic equilibrium occurs. Here, if you see in this figure, this is it. So here they are both sides. This is before osmosis. Here they are said this is selectively permeable membrane. Blue ones are water molecules and yellow ones are sugar molecules. So here before the osmosis, the sugar concentration is greater on the right side. You can see more sugar here. So what will happen because the concentration is more here? So water molecules continue to move across the membrane till the ratio becomes the same. So here more water molecules will enter. After osmosis, once the concentration becomes the same, then the movement will stop and there will be dynamic equilibrium. How many are going on one side? The same many are going on the other side. So this is figure 10, page number 49. Compare and contrast diffusion and osmosis. Diffusion and osmosis both are methods by which substances can be moved. And in both they are moving along the concentration gradient. Diffusion is the movement of particles with a concentration gradient. Osmosis is the diffusion of water across a permeable membrane. So there it is only water and permeable membrane. This is the same thing which I just told you. How osmosis works, I just told you about this. This will come here, one in question number 9 and the other in. So you'll get 2 questions on osmosis. Same thing. Then we come to the 3 swelling of the cells in living organ when put in water. When we put a living cell in water, why does it swell up? Why does it shrink when you put it in a salt solution? So we have to know about from this text. Isotonic solution. Firstly, when a cell is in a solution that has the same concentration of water and solutes, iron, sugars, proteins, and substances as its cytoplasm, the cell is said to be an isotone. So when the cell is in a solution which has the same concentration as the cytoplasm. Cytoplasm is inside the cell and the solution outside has the same concentration as the cytoplasm. We say that it is an isotonic solution. Iso means equal, same. Water will move still through the plasma membrane, but the water that is entering, the same amount of water is going to leave. It will enter and leave at the same rate. So no net movement. No net movement of water. It is moving, but there is no net movement. The cell is at equilibrium. We say it is at equilibrium and there is no net movement of water. The cells retain and the cells will not change shape. They will retain their normal shape. Most cells in organisms, they are in isotonic solutions like blood. Blood is isotonic. It has the same concentration as our cytoplasm. Then when they're in hypotonic, hypo means that the concentration of the solution is less than the concentration of cytoplasm. Now outside it is less and inside it is more. So what will happen? It will enter. Water is going to enter, just like in the previous tube one you saw. Water will keep entering till the solution becomes the same. As a result, osmosis, the net movement of water through the plasma membrane is inside into the cell. Pressure generated as water flows through the plasma membrane is called osmotic pressure. The pressure is called osmotic pressure. In an animal cell, as water moves into the cell, the pressure increases and the plasma membrane swells. It will keep swelling, swelling, swelling. If the solution is extremely hypotonic, the plasma membrane might be unable to withstand and it can burst also if it is extremely hypotonic. But in plant cells, what happens? Because they have rigid cell wall, the plant cells, they do not burst. Animal cell can burst, but not plant cell when they are placed in hypotonic. But as the pressure inside the cell increases, the plant's central vacuole fills with water, pushing the plasma membrane against the cell wall. So it will push against the cell wall, but it will not burst because the cell wall is quite rigid. Instead of bursting, the plant cell becomes firmer. It becomes like fur. That's why in the grocery stores, you see that they keep looking fresh. Fruits and vegetables look fresh because they mixed fruits and vegetables with water. They will not burst. So they will just become like, they look firm. So that's why in the grocery stores, they like mixed fruits. They always keep spraying water over the veggies and the fruits. Now, if they come in a third type of solution, that is hypertonic. Hyper means the concentration outside is more than the concentration inside the cell. Solute outside is the cell is higher. Concentration of solute is higher. Then what will happen? Water from inside will go out because solute outside is more. And when from inside it will start going out, the cell is going to shrivel. It will shrivel because the pressure will decrease. So it will shrivel. And plant cells in hypertonic solution lose water. Plant cell will lose water. They will will shrivel. They'll shrink away from the cell. Loss of water in plant cell causes wilting. So in plant cells, it is called wilting. And go through this information, very important. Here you can see the figures. Sometimes they give you this figure and they ask you question, or they can only give you this figure. So please see that you know the three figures. When you see a cell like this, fully full, like a ball, here this is the original red blood cell is like that. This has been filled with water. Here it has lost water. So what's happening here? I see this figure. Water is entering the cell, but more water. How do you know more water is entering? Because the arrow is broader than this. The arrow, it will go in both directions, but the amount of water that enters is more than the amount of water that leaves. So net movement is inside. It's going into the cell. This is which type of solution? Hypotonic. Why? Because more solute outside. The concentration of solute is, the water concentration is low. In a hypotonic solution, water enters the cell by osmosis, causing the cell to swell. What happens? Cell swells up. Animal cells may continue to swell until they can burst, but plant cells, Because of their cell wall, they will not burst, but the pressure will increase. That's why the grocery stores spray water. Okay, so hypo, the solution outside is less, the concentration outside is less than the concentration inside. Then in isotonic, the concentration outside is equal to the concentration inside the cell. So how much water is going out? The same amount of water is entering. See the size of the arrows. There will be no net change in the cell. Cell will remain in its original shape. You can try to identify somehow these. Sometimes they give these pictures and then students get confused, but it will be the same picture from the book. So try to find, like in this, there's only one on the top. It's hypotonic. Here they've given you 3. This you can identify, but this one is like this and this are similar. So here you can see this is like 3 here, or you can see the number of cells is more here and here it is less, shown in this figure. Okay, so the isotonic solution, water molecules move into and out of the cell at the same rate. Rate is same. And the cells retain their normal shape. The animal cell and the plant cell have their normal shapes in isotonic. Then we come to hypertonic. Hypertonic, the solution concentration outside is more than inside. So what happens? More water goes out. You can see the arrow. More water goes out than enters the cell. And as a result, the cell will shrivel up. It will shrink. It will wilt. Plant cells will wilt. You can see what's happening to the cell inside. It's shrinking. It's in the hypertonic solution, water leaves a cell by osmosis, causing the cell to shrink. Animal cells shrivel up as they lose water. A plant lose internal pressure and they shrink away from the wall, cell wall. So these 3 figures are very important. This was question 22. Then question 28 again is talking about the same thing which I explained you. So there are 2 questions on hypertonic, isotonic, and okay, 28. This is the same I've already explained you. Then question number 23, it's talking about information from text page number 52, that is active transport. Now we already studied that passive transport is when no energy, extra energy is needed for the transport of substances. But sometimes substances must move from a region of low to high, low concentration, that is against the concentration gradient. Concentration gradient is from high to low. But when they're going against the concentration gradient, that is from low to high, low concentration to high concentration, then they need energy. They cannot go like you have to, if you have to move from a lower, like on the hill, from bottom, you want to climb up, you need energy. So they need energy. This is known as active transport. Okay, the movement of substance across the plasma membrane against a concentration gradient requires energy. Therefore, it is called active transport. Here this shows active transport, and this happens with the help of carrier proteins. Carrier protein helps it. Commonly called pumps. So when they are transporting substance against the concentration gradient, we call them pumps. They are called pumps. Some pumps move one type of substance in only one direction. Other pumps move 2 substances across the membrane in the same direction or in opposite direction. Because of active transport, the cell maintains proper balance of substances which it needs. Why active transport? So that there can be a balance. Active transport helps maintain homeostasis. So it's mainly used for maintaining homeostasis. Here they are showing you where only in one direction it is moving. Solute is coming here from low to high and it goes in. It comes here, then it opens, then it goes in. This is requiring energy. But they will talk to you more about this has been a sodium potassium ATPase pump. What is pump means active transport. Pump means needs energy. And how it uses which proteins? Carrier proteins. So the sodium potassium ATPase pump shown here is an active transport pump found in the plasma membrane of animal cells. What's happening? You have to remember the order of the steps that are happening in the plasma membrane for this pump. First step, 3 sodium ions, sodium positive, 3 sodium ions from inside. Inside there is less sodium, outside there is more sodium. So they're moving from less to more, higher, lower to higher. They need active transport. So 3 sodium, they go and they attach to the 3 grooves on the carrier protein. So here you see the 3 sodium ions are attaching here. Second step, the ATP comes because it needs energy. Then only it is going to go. So ATP, it attaches to the protein with the where the sodium has already binded. So it is sodium bound. So ATP attaches. Now when ATP attaches, it will give energy. The breakdown of ATP to ADP, energy is given. ATP breaks to ADP, energy is given. It causes the shape to change. Because of energy, the shape changes. And when the shape changes in the protein, its sodium goes out. Sodium ions, they leave. So 3 ions are going outside. Do you understand these 3 steps? 3 ions attach. They bind with the carrier protein. Once they bind with the carrier protein, ATP, it attaches to the protein, gives energy by changing into ADP, and because of this energy, the protein changes shape and sodium ions go out. Clear? Next step after these 3 sodium. Now we come to 3 for potassium. Now the potassium from outside have to go in. Only 2 potassium ions will bind to the other side here. There are groups for only 2. So 2 potassium will bind from outside. Here they are bind. Once they bind, the potassium, it releases phosphate. The phosphate which was here, it releases the phosphate from the protein and the shape changes back. When the shape changes back, the potassium comes in. It comes to its original shape and the potassium moves. So these are the 6 steps, very important. How many ions are going out? Sodium, 3 ions are going out. How many potassium are coming in? 2 potassium. 3 going out, 2 coming in. Okay. Then the pump maintains a level of sodium ions and potassium ions inside and outside the cell. The protein pump is an enzyme and catalyzes the breakdown of an energy storing molecule. The pump uses the energy to transport 3 sodium ions out of the cell while moving 2 potassium ions into the cell. 3 are going in, 3 are going out, and 2 are coming in. The high level of sodium on the outside of the cell creates a concentration gradient. Now, there are too many sodium here. There is a concentration gradient. So what happens? The activity of sodium pump can result in yet another form of cellular transport. Substances such as sugar molecules must come into the cell from the outside. Sugar is needed in the cell. So sugar has to come inside from the outside where the concentration of the substance is lower than its inside. So outside there is more sugar, inside there is less sugar, it has to come in. This requires energy. It requires energy. So this requires energy. Recall that potassium ATPase pump moves sodium out of the cell port. The sodium ions that have been pumped out of the cell can couple with sugar molecules because 3 are going out, 2 are coming in. So they couple with this. 1 will couple with a sugar molecule, and this coupled with this, it can enter cell by facilitated diffusion. So sugar will move with the sodium, and it will come by facilitated diffusion of the sodium. As a result, sugar enters the cell without spending any energy. This type of transport occurs in some plant tissues also. Sugar moves by coupled transport into plant tissues. These tissues carry the sugar to parts of the plant where it is stored and until it is needed. So this is very important. Like piggyback, their way into the out of the cell by coupling with another substance using active transport. So that's it. Now there are 2 more processes which you should know from here, endocytosis and exocytosis. These both are active transport. Endocytosis means something entering into the cell, and exocytosis is something going out of the cell. It is a process by which surrounds a substance in the outside environment, enclosing the substance in a portion of the plasma membrane. We'll come to endocytosis. This I've already explained to you. to you. This will also come in question number 24. So this I have already explained to you. All this I have explained to you. Now this is endocytosis. Endocytosis, what's happening? Endo means coming in. There is supposing a protein outside. Now it has to come in. Or food outside, like in amoeba. Amoeba takes in food with the help of endocytosis. So the last substance, it enters the cell by endocytosis. It is engulfed and enclosed by a portion of the cell's plasma membrane. Plasma membrane sinks and substance is enclosed by the plasma membrane and it forms a vacuole. It forms a vacuole. The contents move inside the cell. This is endocytosis. Exocytosis is the opposite of endo. It means the substance has to leave the cell. It has to go out of the cell, like waste material has to move out of the cell. Cells use exocytosis to expel waste and also to secrete substances like hormones, which have to go out of the cell. Both endocytosis and exocytosis, they require energy, so they are part of active transport, and they maintain homeostasis by moving substances into and out of the cell. Some transport processes require additional energy input while others do not. Together, the different types of transport allow a cell to interact with it. Now here you can see that for endocytosis, a vesicle is formed. All the waste collects into the vesicle. Here was vacuole, here is vesicle, and this vesicle goes and fuses, joins with the plasma membrane, and it throws the contents out of the blood. So this is exocytosis. So this is all in this lesson. Then we come to the next lesson, which is organelles and structures. So figure 18, page 57. This is what cytoskeleton. So they will ask you about cytoskeleton. What are cytoskeleton? Cell biologists have discovered that organelles do not float freely. First, it was thought that the organelles, they are floating freely in the cell. But now scientists know that they are not floating. They are in their positions and they are held by a special structures which are called which is cytoskeleton, which is holding them, and they do not float freely in the cell. The organelles are supported by a structure within the cytoplasm, which is just like this structure. We call it the cytoskeleton. It is a supporting network and it has two types of protein fibers. Fibers are made up of protein, two types of items. One is long, thin protein fibers like these ones, like long and thin protein fibers, and forms a framework of this for the cell and provide anchoring for the organelles inside. They anchor the organelles, they hold the organelles. The cytoskeleton also has a function of cell movement and other cellular activities. Now, cytoskeleton is made up of two types of tubules or two types of structures, which are called microtubules and microfilaments. Microtubules are long hollow protein cylinders. These are long hollow protein cylinders. These ones, they are microtubules. They form a rigid skeleton for the cell and assist in moving substances within the cell. Inside the cell, they help in moving substances. Microfilaments are the yellow ones. These dark yellow or ochre colors. So microfilaments are thin protein threads and help to give shape and enable the entire cell or parts of the cell to move. Microtubules and microfilaments, they assemble and disassemble and slide past one another so the organelles can move. This is a cytoskeleton. Here they said, this is the question number 25. Summarize the structure and function of cytoskeleton. So cytoskeleton is made of microtubules and microfilaments. These protein cylinders and threads give cell shape and help in cell movement. Then next we come to information from text page number 59. This will talk about plant cell, animal cell, eukaryote, prokaryote, the structure and function, cell wall. Firstly, plant cell wall. This is a structure which is associated only with plant cells. It is thick, rigid mesh of fibers that surrounds the outside of the plasma. Inside you have the plasma membrane and outside you have the cell wall. What Robert Hooke saw was a cell wall. So it protects the cell and it gives it support. It's rigid, it's thick, so it is giving support and it's protecting. Rigid cell walls allow plants to stand various heights. So plants, how can they stand so high? So they need some support. That is even from the blades of grass to the big trees, they are providing support. Plant cell walls are made of cellulose. Remember this, that plant cell walls are made up of a carbohydrate. The name is cellulose. It gives the cell wall their inflexible character. They cannot, like, it's inflexible characteristic. So this is about the plant cell. What happens to the prokaryote? Even prokaryote, even some of the bacteria have cell wall. So the prokaryote, such as bacteria, also have cell wall surrounding the cell. The function of a prokaryotic cell wall are similar to the function of the plant. They protect. It has in protection to make it maintain a shape and give support to the cell. However, they are different because prokaryotic cell walls are not made of cellulose like plant cells. They are made up of another material which is called peptidoglycan. It is made up of another material, peptidoglycan. This is a combination of peptide and disaccharide fragments. So all prokaryotes have peptidoglycan in their cell walls, and plant cells have cellulose in their cell. So cell wall, plant cells have cell wall and chloroplast. Animal cells have these centriole cilia lysosome. These are differences between plant cell and animal cells. Both have cytoskeleton. Both have endoplasmic reticulum. Both have Golgi apparatus. Mitochondrion, nucleus, plasma membrane, ribosome. This is present in both. This is how they are different. This I already told you. This will also come in another question. Okay, this I've already told you about prokaryote cell wall made of peptidoglycan. Then we come to the next question about page number 62 about ribosomes and endoplasmic reticulum. What are ribosomes? Ribosomes, they are organelles which produce proteins. They produce proteins. They help in manufacture proteins. They're called ribosomes. These organelles are the protein factory. Ribosomes are made up of two components, RNA and protein. So they have RNA and protein. And one special feature of these ribosomes is that they do not have a membrane. Like all organelles have a membrane around them. They don't have an organelle. They do not have a membrane around them. And where are they made? Ribosomes are made in the nucleolus, inside the nucleus. So inside the nucleolus is making ribosome. And then this ribosome is producing proteins. Cells have many ribosomes that produce a variety of proteins that are used by the cell or are moved out and used by other cells. So the cell can use the proteins or they can go out and they can be used by other cells. Some ribosomes float freely in the cytoplasm. Some we just find floating freely, and some are attached to the endoplasmic reticulum. They are bound to the endoplasmic reticulum. Free-floating ribosomes produce proteins for use within the cytoplasm. So those that are floating freely, they are making protein which the cytoplasm is using. And the one which are binding with endoplasmic reticulum, they produce proteins that will be used by other cells. Do you understand that? So free-floating proteins are being used by the free-floating ribosomes are producing proteins which is used by the cell cytoplasm. But the ones which are attached in endoplasmic reticulum, they are used by other cells. What is endoplasmic reticulum? It's also an organelle, endoplasmic reticulum. We also call it ER, E for endoplasmic and R for reticulum. It is membrane system of folded sacs. Here you can see this. This is endoplasmic reticulum. Folded sacs, interconnected channels. And it serves as a site for protein and lipid synthesis. Two things are made in endoplasmic reticulum, protein and lipid. Protein and lipid. The structure of the endoplasmic reticulum with many pleats and folds provides large amount of surface area so that this function can take place. So it's like folded and it has a large surface area. There are two types of ER, rough endoplasmic reticulum and smooth endoplasmic reticulum. So the area of the ER where ribosomes are attached. See where you see ribosomes, you see these blue dots? Where the ribosomes are attached, they are like bumps on the ER. So this is known as rough endoplasmic reticulum. And where there are no, like here in the second part, where there are no ribosomes attached here, in this one, it is smooth endoplasmic reticulum. So the ribosomes, they are producing proteins, and then it goes to the other cells. Smooth endoplasmic reticulum shows that there are areas of endoplasmic reticulum that do not have ribosomes attached. They are called smooth endoplasmic because of their structure, but it does perform an important function of the cell. The smooth endoplasmic provides membrane surface where a variety of complex carbohydrates and lipids, including phospholipids, are formed. So what is being formed here? Lipids and carbohydrates. Smooth ER in the liver detoxifies harmful substance. It removes harmful substance. In the liver, it's doing this function. So proteins and lipids are being made in the endoplasmic reticulum. Then the next question will be from lysosome, page number 64. In the animal cell, there are special organelles which are called lysosomes. Lysosome factories and cells also need cleanup crews. So factories and cells, they want to clean up. Now, who's doing the cleaning up? The lysosome. They are vesicles that contain substances that digest excess or worn out organelles and food. If there are excess particles, worn out organelles, some extra food particles, they cannot keep on accumulating inside the cell. They have to be cleaned up. So the lysosome is cleaning up because it has substances that can digest it. Lysosome digests bacteria also. They can digest bacteria, viruses that are coming into the cell. The membrane surrounding a lysosome prevents the digestive enzymes inside from destroying the cell. So they are surrounded by a membrane, so they do not destroy their own cell. They will only destroy the substances which are not needed by the cell. They can fuse with vacuoles and dispense. So they go and they attach to the vacuole. They give the digestive enzymes in and they digest the substance. They digest the waste. So they have digestive enzymes inside the lysosome. On the next page, summarizes, compares, and contrasts the general structures of plant and animal cells. Notice that plant cells contain chlorophyll. I've already told you they capture and transform energy from sun into usable form of chemical energy. In addition, animal cells usually do not contain vacuoles. Plant cells have vacuoles, but animals don't. If they do, vacuoles in animal cells are much smaller than in plant cells. They do not have cell walls. Cell walls give plant protection and support. There are many structures found in both plant and animals, as we had discussed earlier. Okay, so this is about lysosome. I already showed to you. This is a TEM magnification, 13,500 times. Transmission electron microscope. What is the role of endoplasmic reticulum? Question number 30. The rough endoplasmic reticulum has ribosomes that produce proteins for export. So rough has, because it has ribosomes, it produces proteins and they go out of the cell. Smooth endoplasmic reticulum is involved in the synthesis of complex carbohydrates and lipids. So this is the function of the endoplasmic reticulum. Now here they have shown you question number 27. They said will be from this table. So they made it like a very broad thing. You should know the functions of all the structures and where we find them. Like cell wall, we've already discussed. Centrioles are organelles that, they are formed by microtubules. Many microtubules will join together and form the centriole. It's only found in animal cells and they are important for cell division. Then chloroplast, very important. We'll come to chloroplast later. They are found only in plant cells. They help in the process of photosynthesis. Cilia, they are projections from the cell surface. They help in locomotion they help in movement of unicellular organisms, and also they sweep to remove substances along the surfaces found in animal cells, protist prokaryotes. Cytoskeleton we've discussed. Endoplasmic reticulum we've discussed. Flagella also like cilia. They're also used for locomotion and feeding. They are longer. They can be more than one found. They can be like one or two found in the cell. Cilia are smaller and they are more in number. They are also found in some plant cells. Then Golgi apparatus, we will be studying about it later. It has like many sac-like tubular membrane, and it packages and distributes the proteins inside. It just packages and sends them out. Lysosome we've discussed. Mitochondria, the powerhouse of the cell, membrane-bound organelle. It makes energy, found in all eukaryotic cells. Nucleus is found inside inside the cell, and it has this inside is nucleolus. This whole thing is the nucleus. It is the control center of the cell. It contains coded directions for the production of protein. It has genetic information. Plasma membrane we've discussed. Ribosome we've discussed. And vacuole is a membrane-bound vesicle for storage of material. Especially big vacuoles are found on plant cells. In animal, they are not found. If they are found, they are very small in number. Now, organelles at work. With the basic understanding of the structures found within a cell, it becomes easier to envision how these structures work together to perform the functions. For example, the synthesis of protein. Protein synthesis begins where? In the nucleus. With the information contained, the DNA has the information. Now, this information from the DNA is copied and transferred to RNA. RNA and ribosomes, which have been manufactured in the nucleolus, which are formed in the nucleolus, they together will manufacture proteins. Each protein made on rough ER has a particular function. It might become a protein that forms the part of plasma membrane or will go outside the cell for other cells. Okay. Then other ribosomes will float freely in the cytoplasm and also make proteins. Then proteins made on the surface of the ER are sent to the Golgi apparatus. The Golgi apparatus is going to package them and put them into small vesicles. These vesicles will transport the proteins to other organelles or out of the cell. Other organelles will use these proteins to carry out their cell processes. Lysosome use enzymes to digest food and waste. Mitochondria use them to produce energy. So all this we see that they are working together like a factory, different departments, and then they are working together. Okay, so that is all in this. Then we come to the next lesson, which is about plants. Table 4, page 81. This table and this. So what in plant cell, by presence of a cell wall, a large central vacuole and chloroplast. You know that plant cell have these things. There are many different types of plant cells which carry on different types of adaptation, and then they join together to make different types of tissues in a plant cell. 3 types of plant cells are forming different plant tissues. These are the 3 types of plant cells. You should know the 3 cells. First one is parenchyma, second collenchyma, third sclerenchyma. Parenchyma, see the images. You should remember the images as well as the functions. So parenchyma function is it has like many functions. Storage, firstly, it stores. Then photosynthesis is also taking place in parenchyma. Gas exchange is taking place in parenchyma. Protection and also tissue repair and replacement. So 5 functions. Then collenchyma has 3 functions. This is the functions of collenchyma. In your book, they have switched it. So please see, be careful. What does collenchyma do? It supports support for surrounding tissue. It provides flexibility for plants. So that is flexible. It can withstand different weather conditions. It also repairs and replaces the tissue. Then sclerenchyma is a dead tissue. When it becomes mature, it is not living, it is dead. Sclerenchyma, it's a dead tissue. And its function is to provide support and transport of materials. That's it. So girls, I think we are done with it. I know you will be amazing. Please go through all the lessons, through the practice questions which I have given you, and definitely you are going to do good. It's multiple choice questions. I understand 30 questions. You have to manage your time. Read the question carefully, not one time and answer. Read it 2 times, 3 times. And then sometimes, there's a lot of reading. So read it carefully and then answer the questions. And I know you all are going to do very well. Good luck. And I want you to come out from your exam hall with smiling faces. Thank you. And once again, good luck.